Chapter 7, the nervous system. We've skipped Chapter 6 on the skeletal and muscular system simply because I don't have enough time to cover it, so here we are at Chapter 7. Here are the primary objectives for this chapter. I'll just go ahead and let you read these yourself this time instead of reading them for you. And I'll just hold the video here for a while so you can look at them. Okay, moving on. Section 7.1. The nervous system is categorized by structure and function. And now we're going to talk about the two basic components of the nervous system. There's the peripheral nervous system, or PNS. More about that in a second. And then there's the central nervous system, or CNS. And this includes the brain and the spinal cord, both of which are encased in the axial skeleton and are covered by what are actually three membranes, the meninges. And the central nervous system is the main integration center of the body, which we're going to say here means it just analyzes the incoming information, which leads to the generation or creation of a motor response, which is usually directed at muscular or glandular or endocrine system tissue. And then the PNS is simply all of the nerves and other structures towards the lateral parts of the body. And I'm just going to use the word lateral here because we just learned that word. So it's the PNS is the lateral part of the nervous system, whereas the CNS is the centrally located portion of the nervous system, and that's the main difference between the two. The peripheral nervous system connects to the central nervous system, and a more accurate way to state what I said in the last slide would be just to say that the peripheral nervous system is composed of all the afferent and efferent neurons that extend from the central nervous system. So it just includes all the neurons that carry information to and from the central nervous system. And like I said, the nervous information or nerve impulses are carried by the neurons or cells of the nervous system. And this information is passed from cell to cell using neurotransmitters, which are the chemical messengers of the nervous system. And we'll talk more about where and when neurotransmitters are used in the following slides. And the neurons of the peripheral nervous system, not the central nervous system, are arranged in bundles which are called nerves. And these nerves of the peripheral nervous system can be classified as motor, sensory, or mixed. And mixed simply means that these neurons could be carrying either motor or sensory information. Nearly all information going to and away from the CNS must travel through the PNS first. And this information reaches the CNS from the afferent division of the peripheral nervous system. And now we're going to talk about what the difference is between afferent and efferent. So afferent is a word used to mean that we're going towards something, whereas efferent is a word used to mean that we're going away from something. So here we use the word afferent to signify that the information is going towards the CNS, and we use the word efferent to signify that the information is going away from the CNS to the motor neurons of the peripheral nervous system. And the information which the PNS picks up is gathered through the use of one of three types of receptors. So we have the receptors of the special senses, or special sensory receptors. Then we have general sensory receptors, and we also have visceral receptors. So the special sensory receptors enable us to see, hear, taste, and smell the incoming information from the external world. So these receptors would be found in the eyes, nose, ears, and on the tongue. The general sensory receptors inform us about general information such as the relative external temperature, as well as other changes to our environment or other stimulation. So they would sense things like light touch, a change in pressure, and also pain. Finally, the visceral receptors monitor proprioception and organ functioning. So the word proprioception is used to mean that these receptors help us to realize where our body parts are in relation to the rest of our body. So if we were to say raise our hand or something, these receptors would tell the brain that our hand is raised and is higher than it would normally be. And the organ functioning that these receptors would be monitoring would be something like stomach aches and sore throats, and these are examples of visceral sensory input. The peripheral nervous system consists of the somatic nervous system and the autonomic nervous system. And before we move on with this, I'm just going to say that remember that the central nervous system has no further divisions. It's the central nervous system and that's it. Whereas the peripheral nervous system has these two divisions, and then the autonomic portion of the PNS is then divided into two divisions itself. So the somatic nervous system of the PNS is what is sometimes called the voluntary division, because this is where the motor commands are consciously sent. So we have to send a conscious thought in order to work this part of the nervous system. However, it does include the involuntary movements produced by our reflexes. So maybe a better way to describe this division is that it's the portion of the nervous system in charge of our skeletal muscles. The autonomic nervous system is the control system which governs our body's responses to slight changes in homeostasis and results in involuntary unconscious reactions. So this is the part of the nervous system which would control your heartbeat and the functions of your lungs, and also digestion and some other things, obviously. And this is made up of two divisions, the sympathetic division and the parasympathetic division, which are on the next slide. The sympathetic division of the autonomic nervous system, or ANS, is sometimes referred to as the fight or flight division because it's triggered when we feel threatened or when the fight or flight response is induced. And this division still includes those nerves which control the body when it is actively moving or burning energy. So when this division of the ANS is activated, it would cause your heart rate to rise, your blood pressure to rise, your ability to take in air in your lungs would increase, and you'd also begin to feel that feeling of being threatened or that you're afraid. And so a good way to remember this part of the ANS is to remember that the sympathetic division is sympathetic to your plight or whatever is threatening you. In contrast, the parasympathetic division is that part of the ANS which is responsible for your digestion, energy storage, and it's active when you're relaxed. And this is in contrast to the sympathetic division, which, yes, still has controls in the digestive system, but generally the sympathetic division leads to the shutting down or decreased digestion, whereas the parasympathetic division, since it's active when we're at rest, causes our digestive rate to go up and our energy storage abilities to go up. And a good way to remember this part of the ANS is that it starts with P, like potato, and is active when you are relaxing, thus causing you to act like a couch potato. Maybe that's not the best way to remember it, but that's what the book says. Here's a flow chart of the events that occur in the central nervous system, and this would be really good for you to study and be able to separate the various divisions. So in the somatic division, the information goes to the central nervous system, as does any sensory information. And then the central nervous system sends this information to the somatic neurons, which are found in skeletal muscle. And contrast this with the autonomic nervous system, which is divided into the sympathetic and parasympathetic divisions. But anyway, the information from the ANS travels to the CNS, where the information is processed and then sent to the atomic motor neurons of the sympathetic and parasympathetic divisions. And these neurons are found in the smooth muscle, the cardiac muscle, and in the glands. Nervous tissue is made up of neurons and glial cells, and I'm going to try to just call them glial cells because I have the tendency to get tongue-tied when I say neuroglia. Anyway, the glial cells are the cells of the nervous system which provide support and protection to the neurons within the nervous system. And these include cells which provide nutrients to the neurons, cells which remove debris from the nervous system, and cells which help to speed up impulse transmission of this neuronal information. More on glial cells in a second. Neurons come in three different types. There are sensory neurons, which send sensory information to the brain. There's the interneurons, which are somewhere in between the sensory neurons and the motor neurons. And these can be either afferent or efferent. And then there's the motor neurons, which are efferent neurons, and these are the neurons which receive the information from the brain. However, all three types are somewhat similar, or very similar rather, as they all have one cell body, one axon, and at least one dendrite. But all of them have a distinct shape related to their function. Here's a picture depicting motor neurons, interneurons, and sensory neurons, and I'm not going to address why each of these types of neurons is shaped like it is. But anyway, just know that the neuron at the far left is a motor neuron, the neuron in the middle is an interneuron, and the one at the far right is a sensory neuron. Here's a chart of the 6 types of neuroglial cells in your book, and I'm not going to need you to memorize all of this or anything, but I need you to be aware of a couple things. First, it's probably a good idea to know what a Schwann cell is. This is a cell which surrounds axons in the peripheral nervous system, causing myelination of axons, which allows for faster transmission impulse, and these aid in repair after injury also, and we'll address that in a following slide. And then you need to know what an oligodendrite is. These are basically just Schwann cells of the CNS. They surround axons in the CNS and are responsible for myelination and also provide structural support. And it'd probably also help to know what an astrocyte is, because there will be at least one topic where we'll have to know what an astrocyte is. And these are responsible for maintaining the blood-brain barrier, which basically just involves making sure harmful materials don't enter the brain. They also regulate nutrient, ion, and dissolved gas concentration, absorb and recycle neurotransmitters, and form scar tissue after injury to the central nervous system. Section 7.2, neurons work through action potentials. And before I tell you what an action potential is, we must first know what a membrane potential is. Anyway, a membrane potential is the difference in electrical charge on either side of a membrane. In the case of a neuron, this is a plasma membrane. So an action potential is any dramatic change in charge on on the inside of the neuron, which results in that neuron firing or sending a nerve impulse down its axon. Anyway, when a neuron's at rest, it has a membrane potential of negative 70 millivolts. And in order for us to have this membrane potential, the levels of positive potassium ions and negative chloride ions must be higher outside the neuron than inside. And we must have the more positive potassium ions more concentrated in the inside of the neuron as opposed to the outside. Large negatively charged proteins trapped in the neuron also help to maintain the negative charge across the membrane. As with any other type of plasma membrane, it's selectively permeable, which prevents diffusion, helping to maintain a resting membrane potential. So how do we change the resting membrane potential? We do this using protein channels, which allow ions to travel from inside to outside or outside to inside the neuron. And there are two different types of channels. Passive channels are what we call leaky because they allow a constant trickle of ions into or out of the cell, and these aren't responsible for action potentials. And then the active channel proteins allow no ion movement unless they're stimulated to open, which would cause neurons to fire in this case. The rate of ion movement across the nerve cell membrane depends on the physical state of the channel proteins. So variation in ion concentration across the cell membrane allows neurons to generate action potentials, like I said. This is a picture depicting membrane potential, which pretty much just shows the state or the charge state of both sides of the membrane when the neuron's at rest. So here we have this little graph down here that shows the resting state of the membrane potential is negative 70 millivolts. So that means that we have a pretty negative inside compared to a pretty positive outside. And over here at the farther right is just a close-up of this difference in charges across the membrane. More about active channels, which are also called gated channels, because they allow ion transport only under specific environmental conditions. And there are three different types of gated channels. There are voltage gated channels, which open and close in response to transmembrane voltage changes. There's ligand gated or chemically regulated channels, which open and close when they come in contact with the proper chemical, which then binds to them. And then it can be mechanically regulated, which is a response to physical distortions of the membrane surface. So, like we said in previous chapters, the membrane of cells is pretty dynamic and movable. So these channels would be opened like when the cell's under more pressure than it normally would be. And at rest, these gated channels are always closed. But when they open, the gates allow the ions to cross the membrane, which is regulated by concentration gradients, so always higher concentration to low concentration. And this changes the transmembrane potential, or just membrane potential, and this generates an action potential, or nerve impulse is another name for action potential. Here's a picture of two different types of gated channels, voltage gated channels and ligand gated channels. And in this voltage gated channel we have a sodium potassium, or we have a sodium and a potassium channel. And these are usually the channels that we talk about when we're talking about the creation of an action potential. And in these channels, the sodium moves out of the membrane and the potassium moves in, and this allows for a change in the membrane potential to where it's more positive as compared to its typical negative. And then we have ligand gated sodium channels, and as you can see with this channel, the chemical that binds it and causes it to open is acetylcholine. And in the case of this channel, when it binds this chemical or ligand, it causes the channel to open and sodium flows into the cell. Here's a picture which shows how action potentials are created or propagated. And just to clear up what I said on the last slide about these channels, usually this is referred to as one channel, the sodium-potassium channel, and not as two separate channels. So I guess I just need to clear that up. Anyway, at the resting state of a neuron, we're at our usual negative 70 millivolts, where there's more sodium outside of the cell than there is inside, and there's more potassium inside than outside. And we maintain this negative 70 millivolts membrane potential when both these gates are inactive and closed. However, when these channels open, we start a process called depolarization, in which the membrane becomes much more positive than it normally is, reaching all the way up to positive 30 millivolts. So anyway, when the channels open, the sodium, being slightly more positive than the potassium as far as its charge goes, flows in and helps to make the membrane potential more positive. And at this point, the potassium channels are still closed, but the depolarization of the membrane begins to open more sodium channels. Moving on to the repolarizing phase, and this would occur after we've actually created our action potential. So the action potential has already been created and the neuron should have fired by now. But anyway, to repolarize the membrane, the sodium channels or gates actually inactivate and close, and it's at this point that the potassium channels open, and as the potassium flows out, we experience a repolarization of the membrane back to our normal 70 or negative 70 millivolts. And now the repolarization continues until the inflow of potassium restores the resting potential, and we actually kind of overshoot into a much too negative state slightly here, such that the sodium channels actually have to open up a little, or like a few of them have to open up, to restore the resting state when these potassium gates close. And I hope that's as clear as I can make it, because honestly, this stuff can get a little confusing sometimes. And I'm pretty much just going to ask you, what causes depolarization, or what happens when a membrane is depolarized, and what happens when it's repolarized, and what causes overshoot when we get a membrane potential which is much more negative than what's desired. And we'll talk more about this overshoot in a second. Though all neurons are quite similar, they can propagate action potentials at different speeds. However, all of these impulses always begin at the swollen base of the axon, which is called the axon helix. And the reason why propagation speed can be different depends on at least two different factors. The diameter of the axon. Thicker axons propagate nerve impulses faster than thinner ones, and the amount of a chemical or substance called myelin wrapped around the axons. And myelin is actually made up mostly of white lipids and phospholipids wrapped around the axon, so it's kind of like the insulation wrapped around an electrical wire. And when we wrap this myelin around the axon, we don't connect all of the sheets together. Instead, there's these little nodes between the sheets, which are called nodes of Ranvier, I believe is how it's pronounced. And these nodes are actually stretches of naked axons between the myelin sheets. And myelin is obviously created or produced by either Schwann cells or oligodendrites in the central nervous system. And with these axons, the voltage-gated channels are actually concentrated at the nodes, such that we create APs at these nodes, which then don't have to be repropagated as the impulse moves down the axon. Whereas with a naked neuron, we have to keep propagating this action potential as we go down the axon, which is very time consuming compared to just propagating an action potential very few times, which then moves straight down the axon. Here's a picture of impulse conduction, which ought to clear up a few of the things I just said. So here on the left we have a naked axon. And as you can see, we have a change in the membrane potential when the sodium ions move in, such that the membrane potential becomes more positive. And then we have a little bit of an action potential which just travels a little ways. And then we have to repropagate this every little distance the action potential travels to keep up the nerve impulse and keep it going. And compare this to what's at right, where we have the Schwann cells and the nodes of Ranvier created between the myelin sheets. But anyway, here we can propagate the action potential at the nodes, shoot it through the axon that's covered in the myelin sheet, and then we only have to repropagate them at the nodes, and this is much faster than what's going on at the left with the naked neuron. We're going to talk about Schwann cells and oligodendrites again, just to reestablish the information provided when we went over the chart of the glial cells. Anyway, Schwann cells are found in the peripheral nervous system only, and are the neuroglial cells responsible for myelination or creation of myelin sheaths which wrap around the axon, covering it in phospholipids, which help to increase the speed impulses are conducted. And they also aid in regeneration of neural axons if they're damaged. Oligodendrites, on the other hand, are basically the same as Schwann cells. They're just found in the brain and spinal cord. And these are large cells with branching appendages that touch and protect many axons. They do not aid in axonal regrowth, And this is one reason spinal injuries and brain injuries can be permanent, simply because we don't have this regrowth. And over here at your right, you can see a picture of Schwann cells and myelin sheaths. And don't worry so much about part B on the farther right. Neurons in either the PNS nor the CNS can regenerate if the body cell is damaged. Axons only regenerate if they are damaged beyond the axon hillock, and this explains why there's not really much recovery from something like paralysis. Synapses separate one neuron from another, and neurotransmitters bridge the gap. So now we're going to talk about what happens when neural impulses must jump from axon to axon. So the action potential impulse must be transferred to the next neuron at the terminal bulb or swollen end of the axon, and each neuron is separated from the other neuron by a gap called a synapse. So in order to bridge this gap, neurons must release a chemical messenger called a synapse from the terminal bulb. And in order for the neurotransmitter to get to the receiving neuron, they must traverse the space called the synaptic cleft, which they do by simple diffusion, and that's how they are delivered to the receiving neuron. And the neuron which is sending the neurotransmitter is called a presynaptic neuron because it's placed before the synapse, and then the neuron which receives the neurotransmitter is called the postsynaptic neuron because it's placed after the synapse. Here's a picture which shows what happens at the synapses. So first, the action potential must be received in the terminal bulb, and this leads to the opening of sodium channels, and the resulting depolarization, in this case, causes calcium channels to open. Or I shouldn't say in this case, this is just what happens every time we have to send a neurotransmitter to a neuron. The calcium then causes synaptic vesicles, or just vesicles within the synapse of the neuron, to fuse with the membrane of the neuron and dump the neurotransmitter they contain, in this case acetylcholine, into the synapse. The acetylcholine then transverses the synaptic cleft and then binds to ligand-gated channels. So here we have a sort of indirect interplay between the two types of channels, and this causes the receptors to open, and this lets sodium enter this postsynaptic neuron and cause depolarization of the membrane, and this depolarization of course results in an action potential. So just backtracking a little, just remember that depolarization of a neuron cell is what actually causes an action potential, and this has nothing to do with repolarization of any membranes. So just depolarization leads to action potential. There are several different types of neurotransmitter, and each of these has a slightly different effect on the postsynaptic neuron and plays a different role in the nervous system, but they all carry messages across the synapse. And there are more than 30 or 45 identified neurotransmitters. We're just going to focus on 3. So the neurotransmitter from the picture earlier is acetylcholine, and this is usually just a neurotransmitter used to stimulate muscle contractions, among other things. Then there's epinephrine, which is actually what Americans are supposed to call adrenaline. Adrenaline is actually a word that the British use for epinephrine. And anyway, this increases heart rate and respiratory rate and decreases digestive rate and also provides a burst of energy. Now in your book, they actually say that the effects of epinephrine are not all that well known. This is actually not true because there's been thousands of studies involving adrenaline. Anyway, a chemical or neurotransmitter very similar to epinephrine is norepinephrine, which is also called noradrenaline, and these are related chemicals. And this is similar or has a similar effect as adrenaline because of their structural similarity. Here's a list of some of the neurotransmitters and what they do. And I don't know what's with the author of our textbook because adrenaline's been pretty well classified and studied, so I don't really understand why they wanted to say its effects are uncertain. But anyway, this is a good time to point out that textbooks aren't really peer-reviewed like research journal articles are. So that kind of explains why there's a lot of just not good information in textbooks. But anyway, here's one example. But anyway, just focus on the three that we talked about: acetylcholine, epinephrine. Don't say its effects are uncertain because they are, and also study epinephrine, or at least know sort of what it does compared to epinephrine. So the firing of an action potential is what's called an all or nothing event, because once we reach the threshold for an action potential to be fired, the nerve fires the action potential. So just think of what happens when you fire a gun, which is you don't just sort of fire a gun. Either the bullet comes out of the barrel of the gun or it doesn't at all and it's not fired. That being said, we can vary the strength of nervous stimulation by changing the number of neurons that are firing. And this is how we get a graded response. Graded responses are obtained by either hyperpolarizing or depolarizing individual neuron membranes. And hyperpolarizing is actually what happens when we get a more negative membrane potential than what we normally want, as opposed to depolarizing where it's more positive. A hyperpolarized neuron requires a larger stimulus to reach threshold and begin an action potential because it simply just has a more negative charge here, so we need to get more ions into the cell to reach threshold. And compare this to a more depolarized neuron, which requires a fairly small stimulus to reach threshold simply because we don't need to put so much effort into changing the charge. And hyperpolarization and depolarization of neurons results from alterations in the resting potential of postsynaptic neurons. And there are two types of postsynaptic potential. There's excitatory postsynaptic potential, which causes slight depolarization of the neuron membrane potential, very close to that which is needed to actually generate an action potential, such that a small stimulus will begin an action potential. So that's why they're called excitatory, because they're very excitable. And the opposite of this is inhibitory postsynaptic potentials, which hyperpolarize the neuron such that the membrane potential is farther from that which is needed to generate an action potential, so a much larger stimulus is required to create an action potential. Section 7.3. The brain and spinal cord are central to the nervous system. And at this point we start to move into some easier material. mostly because we're talking about a lot of human anatomy here. The brain occupies approximately 1,250 to 1,400 cubic centimeters and weighs about 1,400 grams. And the brain looks pretty unexciting, but this conceals a great level of detail, and this detail emerges from just a few types of cells, which are specifically and purposefully connected within the brain. Now we're going to talk about the meninges, which are a series of connective tissue coverings between the nervous tissue and the bone of the skull and spinal column, both of which protect the brain and spinal cord. And there are three layers in the meninges. There's the dura mater, which in Latin translates to tough mother, and this is the tough outermost layer of the meninges. Then there's a deeper layer called the arachnoid mater, which translates to spider mother, which is a thin and fragile spider web looking sort of membrane. Then finally there's the P mater or pia mater, which translates to gentle mother, which is an extremely thin innermost layer which is actually attached to the neurons and cannot be peeled off when dissecting a brain without damaging these neurons. And when these membranes become inflamed either by bacteria or viruses, this results in meningitis, which just translates to inflammation of the meninges, which can be fatal and very difficult to treat. And like I said, it can be viral or bacterial, and it's usually viral for the most part, I believe. Cerebral spinal fluid is the fluid within the dorsal cavity which separates the membrane of that cavity from the brain and spinal cord and helps to maintain uniform pressure between the two. And CSF fluid is continuously produced at the chyroid plexus, which is a mat of specialized capillaries in each ventricle, which leaks fluid into the space surrounding the brain, or leaks cerebrospinal fluid. The cerebrospinal fluid is continuously absorbed at the arachnoid villi, which should be somewhere in this diagram, to prevent buildup of pressure because we don't want excess pressure on the brain and spinal cord. And finally, the cerebrospinal fluid helps maintain the blood-brain barrier, making sure that needed nutrients and ions get to the brain and pathogens and debris stay away from it, because we don't want to be getting some sort of brain disease or other problem. Moving on to the brain itself, the brain has four main parts: the brainstem, the diencephalon, the cerebellum, and the cerebrum, which I'm going to cover in the preceding slides. Here are some diagrams of the human brain. So over at the left, there's a colorized brain scan, which pretty much shows only the cerebrum and the cerebellum. And then to the right, it shows the four lobes of the cerebrum: the parietal lobe, the frontal lobe, the occipital lobe, and the temporal lobe, and also the lateral cerebral sulcus, which I'm not sure is considered a lobe or not. But anyway, there's another diagram which has more detail on the actual functions of the various lobes of the brain. Here are some diagram cross sections of the brain. So over here at the left, you can see all four parts of the brain that we talked about earlier: the cerebrum, the cerebellum, the brainstem, and the diencephalon. And then over there to the right is really just a drawing of essentially the same thing. You can see where the pituitary gland is, though. And that's a pretty important gland in humans and many other animals, obviously. The brain stem is the part of the brain which connects to the spinal cord, and it contains vital centers that regulate heart rate, breathing, and blood pressure. And it consists of three parts: the midbrain, the medulla oblongata, and the pons. The medulla oblongata is what controls heart rate and respiratory function, and also blood pressure. It also contains the reflex centers for sneezing, coughing, hiccuping, and swallowing. The motor impulses generated by the higher centers of the brain must travel through the medulla oblongata to get to the peripheral nervous system. And then the pons focuses primarily on respiration, as the only vital center here controls respiratory reflex. And the pons is composed of tracts which carry information up to the brain, down from the brain to the spinal cord, or laterally from the pons to the cerebellum. The pons contains the apnoistic reflex center, which triggers breathing even when the diaphragm, which is a muscle underneath the lungs which helps you to breathe, is consciously held still. It also contains the pneumotaxic center, which prevents the overinflation of the lungs. The second part of the brain, the cerebellum, focuses primarily on muscles and movement, and it looks like a smaller brain hanging off of the back of the rest of our brain. And the cerebellum has two main functions. It helps to maintain muscle tone, posture, balance, and understanding of where the limbs are located, or proprioception, the word we learned earlier. And it's also concerned with fine-tuning conscious and unconscious movements directed by the cerebellum, and thus is important to learning certain motor skills. Such examples of these would be riding a bike, swimming, or learning new information through note-taking, which is pretty important to other kinds of non-motor learning. The diencephalon part of the brain is a relay and integration center for both conscious and unconscious sensory information, and also motor commands. And the centers for visual and auditory startle reflexes are located here. And your book lists three main parts of the diencephalon. There's the thalamus, which serves as a relay station for most incoming sensory stimuli. There's the hypothalamus, which is the part of the brain which secretes hormones which regulate and control the anterior pituitary gland, and also monitor water balance and stimulate smooth muscle contraction. It also regulates our circadian rhythm, or the times when we're awake or asleep. It also regulates body temperature, heart rate, and blood pressure. The diencephalon also contains the limbic system, which is responsible for producing our emotions, and the limbic system contains the hippocampus, the fornix, and the amygdala. And the amygdala is the portion of the brain responsible for fear and anger, mostly. Here's a cross-section of the brain which shows the parts of the limbic system. We won't really cover this much, but just know that the limbic system is the part of our brain responsible for our emotions, and know what the amygdala is, just because it's pretty easy to remember. The cerebellum is your central processing center. This is the part of the brain which processes and integrates incoming information and generates the appropriate response. This part of the brain contacts all other parts of the brain and is the center for higher level thought processes. Now onto the structure. The surface consists of creases called sulci and raised portions called gyri. The thin outer covering of the cerebrum is called the cortex, and this consists of gray matter, while the interior of the cerebrum consists of white matter. And here's the difference between the two. Gray matter contains mainly cell bodies and non-myelinated neural processes, which are the naked axons and dendrites. Connections are made when the axons meet dendrites. In contrast, white matter contains myelinated axons and is white because myelin is white due to all of the lipids and phospholipids contained within it. Information is passed from one area of the brain to the other area via tracts of white matter. Here's a diagram which shows all the functions of the various lobes of the cerebrum, and I need to correct myself from one of the past slides. So this lateral cerebral sulci is just a big crease, I'm assuming along this line here, and it's not a lobe. Sometimes I get confused. So over here to the far left, we have the occipital lobe, which is the area of your brain primarily associated with vision. And then this yellow lobe is called the temporal lobe, and this is the part of your brain associated with your auditory system. And then the parietal lobe is generally concerned with somatosensory information and is a common integration area of the brain. It also contains the part of your brain which integrates gustatory information or information from your taste buds. And then finally in pink is the frontal lobe, which serves as your primary motor area and also contains this spot called Broca's speech area and your prefrontal cortex. And this part of the brain, or at least one thing that it does anyway, is that it's the part of the brain that allows you to maintain your self-control. Now we're going to talk about memory, or rather learning, which is a type of memory. And memory occurs in three distinct phases. First, there's intermediate memory, which is what allows us, or rather prevents us from being bewildered by maintaining incoming information in our consciousness. So this is the first phase, and all incoming information is placed in this phase of memory. Then there's short-term memory, which helps us carry out tasks such as keeping a conversation going or remembering why we are writing a letter, such as like where we are might be another example. Much of this memory is quickly erased, but some of it gets adopted into the long-term memory. And long-term memory is, or can survive for life, or sometimes it fades. But this is actually what we mean when we say memory. Then neuroplasticity refers to the changes which occur when the brain remembers something. When this occurs, specific proteins are synthesized, synapses change in neural pathways, or are rearranged, and changes in dendrites all occur. So we just have a rearrangement of the neurons in our brain when neuroplasticity occurs. So your cerebrum is actually divided into two separate hemispheres, left and right, and both of these are arranged into lobes which carry out specific functions. The cortex of each of these lobes has a motor area, sensory areas, and association areas that integrate new information with stored memories. So we already went over these lobes, but just as a refresher, the occipital lobe is where vision is interpreted, and the frontal lobe is involved in conscious thought processes. And you can go back and look at that diagram just to review. The primary motor area is found in the frontal lobe, and this is what allows you to formulate voluntary motor commands. So this is the part of the brain that you use to decide if you want to walk around or not. Each portion of the body is represented in this primary motor area, and we're going to look at two diagrams called homunculus, which show which parts of the primary motor area control which part of the body. So here are our homunculi, and these show the primary motor area and what portions of this area control what body parts. So the sensory primary motor area is what receives information from these body parts, and then the motor homunculus shows where the information comes from in the brain that controls each of these body parts. And I hope I said that right. But anyway, I can't really look at these without thinking of the bad guys on Full Metal Alchemist, if you're familiar. Sensory information from the skin and skeletal muscles is received in the primary somatosensory area of the cortex, which is located just behind the primary motor area. Sensations from each body part go to a specific segment of this gyrus, and this is the gyrus of the primary somatosensory area. The larger the segment of the primary somatosensory area devoted to each body part, the more sensory receptors are found in that part. And then referred pain is what occurs when any of the nerves along these sensory pathways are stimulated, and then the brain simply interprets these sensations as coming from the organ at the distal end of the pathway, regardless of the source of the stimulus. So basically, this is where you think pain's coming from one part of your body when it's in fact coming from another part of your body that doesn't really have any way of telling you that that part of your body hurts. So this is kind of like what happens when your insides hurt, like your intestine or your stomach. So you'll think that the pain is located in one area when it's in fact coming from another. Now we're going to talk about some more specific areas of the brain which have specific functions. A few specialized actions are governed by areas outside the primary motor or somatosensory areas. So we have Broca's area, which is located on the left frontal lobe, which organizes the formation of words. And then we have Wernicke's area, which is found on the posterior left temporal lobe, and this is responsible for understanding language, including those created by Broca's area. So these two little areas of the brain work together to formulate words and understand language. The two hemispheres themselves are also distinct. So generally, the right hemisphere analyzes sensory input, recognizes faces, and functions in spatial relationships, and emotionally interprets conversation. Then the left hemisphere, this is involved with general language interpretation. It controls writing and speaking, mathematical calculations, categorizing items, and making logical decisions. So I guess you could say that the right hemisphere is mostly responsible for your more creative emotional side, I suppose, and the left hemisphere is more devoted to your logical, organized, mathematical side. And then, like we've said a couple times before, some special information is integrated in specific areas of the cerebral cortex. I don't know what I was trying to say in that sentence, but I think I forgot a word. So the occipital lobe is the part of your brain devoted to vision, the temporal lobe is the part of your brain devoted to hearing, and the parietal lobe is the part of your brain devoted to taste, and probably smell also, simply because taste and smell are two very closely related senses. Association areas of the cerebral cortex integrate and coordinate information from many sources, or we could just say they link information together. Somatosensory association area is the part of the brain which processes sensory information from the skin and muscles. And then visual association areas, so new visual information with stored visual images. So I already said that the brain is divided into two halves, and this is what's called hemispheric lateralization. And this allows for tasks to be isolated to either the right or left hemisphere of the cerebrum. The left and right sides of the brain connect through the transverse tracts of the corpus callosum, and this allows the two sides to share information and generate different responses. The entire cerebrum is aware of incoming sensory information as well as outgoing motor responses, but I'm sure you knew that already. The reticular activating system is the brain's alarm clock. So reticular formation serves as an important connection between the various parts of the brain. A series of nuclei and tracts extends throughout the brain, receiving sensory information, parceling it to the higher integration centers, and then directing motor responses to the appropriate body areas. So let's talk about reticular activating system, or RAS. A portion of the reticular formation which is important in maintaining alertness. And this helps to filter out noise and focus on important information. So this is the part of our brain that allows us to focus no matter how distracted we might be based on the information or the distractions surrounding us. And it is thought that in attention deficit hyperactivity disorder that RAS may not be functioning properly and might be partially causing this disorder. Now we're just going to briefly look at this chart of common mental disorders from your book. So here we have the anxiety disorders, which include phobias, panic disorders, and obsessive compulsive disorders. And I personally nearly have a phobia, and it's of amphibians, but it's not fully developed. I kind of just more hate them than am afraid of them, but I've always been weary of amphibians. Then there's mood disorders such as depression and bipolar disorder. And I was just going to point out with bipolar disorder, because there's some confusion surrounding this disorder. This chart says that it's characterized by violent mood swings, and this should probably say violent changes in mood. And I'm just going to tell you that bipolar disorder doesn't mean that anyone that has it is necessarily violent. I mean, they can be, but it would be more on their own free will, I suppose, is what I should say. Then we have schizophrenia, and I'm going to skip down to these two eating disorders, anorexia nervosa and bulimia, which we'll actually address in the nutrition chapter. And then I had to talk about Alzheimer's, which is a type of dementia generally occurring in the elderly. And this results in loss of mental function and memory followed by a decline in physical abilities, and leads to an increased need for nursing care. And I only point this out because we're going to have a wiki on Alzheimer's where I ask you some opinionated questions about it and then have you answer some questions over a video. The spinal cord is another area of the central nervous system, and it connects to almost everywhere. It extends from the brain into the vertebral column, and the spinal cord consists of white tracts surrounding gray matter, which is the opposite of the brain, which is gray tracts surrounding white matter. The exterior of the spinal cord is composed of communication tracts, which run up and down the spinal cord, while the interior is composed of connections between spinal nerves. And the spinal cord serves as the main communication route between the brain and the rest of the body. Sensory information enters the spinal cord at the dorsal root and is then transferred to an upward tract which heads to the brain. And then in turn, motor impulses which are generated in the brain are then passed through the downward tracts of the spinal cord to the nerves of the body and then to a specific body part. This picture has a transverse sectional of the part of your spinal cord located in the thoracic portion of your vertebral column. So the main takeaway I want you to get from this is that the gray matter is surrounded by white matter in the spinal cord. And also note where the dorsal root is, because that's where neural impulses enter the spinal cord. And then, well, that's in blue. And then in red you can see where the nerve impulses leave the spinal cord through the downward roots of the spinal cord. Now we're going to talk about spinal reflexes, or simply just reflexes. And these are initiated by sensory information which requires immediate attention. And these are extremely quick responses to stimuli which run through the spinal cord from the dorsal root immediately to the ventral root and bypass the brain, just because we need a response, and we need a response right now. Incoming sensory information is transferred to an association neuron in the innermost portion of the spinal cord and then directly to a motor neuron. This allows the motor neuron to transmit an immediate response through the ventral root to the effector organ. And obviously reflexes can generate or do generate an immediate and possibly life-saving motor response in some cases. So here's the reflex arch, which I guess we could just say is simply the pathway the stimuli and then response takes through the spinal cord bypassing the brain. So first, the sensory receptor receives the stimulus. It sends it to the sensory neuron, which sends it to the spinal cord. We bypass the integration center, which is the brain, and then we generate an immediate motor response mediated by the effector neuron. Section 7.4, the peripheral nervous system extends the central nervous system. And I honestly think this should have been in a section earlier, but this is how they wrote the book. And obviously, we've said it several times before, the peripheral nervous system, or PNS, is composed of all the tissue, or all the neural tissue, in the nervous system other than the brain and spinal cord. And it obviously includes all the nerves that protrude from the brain and spinal cord. And there are 12 nerves that extend from the brain and are called the cranial nerves. And a couple of these are sensory only, one is motor only, and the remainder serve both functions. And I don't know why the book didn't simply specify how many of these, because I don't know, the writing's just all wrong in there. Most cranial nerves carry impulses that deal with the head, neck, and facial regions. An exception to this would be cranial nerve 10, or the vagus nerve, which branches to the throat, voice box, and abdominal organs. So here's a diagram showing where all 12 of the cranial nerves are located in the brain. And as you can see, there are 2 sensory neurons, 1 motor, and all the rest are mixed. And I just want to point out the olfactory nerve or bulb here, and this is responsible for smell mostly. And I only point this out because it's a pretty easy nerve to remember, so it's probably a good idea to at least learn that one at the very least. There are 31 pairs of nerves which extend from the spinal cord, and these are called spinal nerves. All of them are mixed nerves, meaning that they carry both sensory and motor information. Each spinal nerve connects with body structures or body parts near the region where it originates. Sensory neurons carry this information to the central nervous system. They join other motor and sensory neural processes to form these spinal nerves. Sensory neurons separate from the motor neurons before they enter the spinal cord. Sensory neurons enter the spinal cord at the back through the dorsal root of the spinal nerve, and the cell bodies are located just outside the CNS in the dorsal ganglia. Motor neurons exit the spinal cord at the front. Their cell bodies are located within the CNS, and their axons extend out through the ventral root of the spinal cord. The motor and sensory neural processes can be very long. The axon of the motor neuron that moves the big toe reaches from the sacral area, or your rear end or pelvis area of the spinal cord, and runs down the entire length of the leg, which can be up to a distance of 1 meter, or about 39 inches if you're not familiar with the metric system. So here's a picture which shows the posterior and anterior views of the areas where the spinal nerves originate in the spinal cord. And obviously we don't need to know any exact names because there's just too many of them. Just know there are 31 pairs. The peripheral nervous system also contains sympathetic and parasympathetic nerves, and as both of these are in the autonomic division of the PNS, they're autonomic nerves, and we can't consciously control them. So when the nerves in the sympathetic division are responsible for quick movements and energy expenditure. The fibers of these nerves extend from the cervical and lumbar spinal cord, or the neck and lower back, to a series of ganglia, which are a group of cell bodies in the peripheral nervous system, and this is called the sympathetic chain and is on either side of the spinal cord. At these ganglia, the neurons from the CNS synapse with a second neuron that extends to the effector organ. The neurons leaving the spinal cord and synapsing in the ganglia are preganglionic. Those that leave the ganglion and synapse with the effector organ are called postganglionic. So here we have the words effector and effector again. So you really need to learn what they mean, or know what they mean. So here's a diagram which shows the sympathetic nerve fibers. I'm not going to test you over this, mostly because it's just too complicated and I can't figure out a way to ask you any questions. But anyway, this is kind of interesting to look at, just seeing what parts of your neurons from or nerves run from the spinal cord to each effector organ. But anyway, it's just something to look at. Moving on to the nerves found in the parasympathetic division. The fibers of these nerves are found only in the cranial and sacral regions of the spinal cord. So the regions of the spinal cord towards your head, and then the regions of the spinal cord towards your pelvis, or in your pelvis. Neurons leave the spinal cord or cranial nerve and join a ganglion near or in the effector organ. And I should say a ganglion is just what neurons are called in the peripheral nervous system. The parasympathetic preganglionic fibers are long, and the postganglionic neurons are extremely short. And of course, since they're in the parasympathetic division, the fibers trigger relaxation, digestion, and energy storage. And I guess I should differentiate between nerves and neurons one last time. So a neuron is just a single cell in the nervous system, and nerves are a group of cells in the nervous system. And usually we apply the term nerve in the peripheral nervous system and not in the central nervous system. So just try to remember that nerves don't really occur in the central nervous system, or at least they're not called that. They're in the peripheral nervous system. So here's a picture showing the parasympathetic nerve fibers, and once again it's really just something interesting to look at because it shows where each of these fibers goes once it leaves the area where it originates. And that's all she wrote for this chapter. In the next module we'll be covering chapter 8, which is over the special senses, and is very much the same sort of stuff. We just cover the organs which allow you to use your special senses and how they are connected to the brain, at least somewhat. But anyway, I was going to try and put the two chapters together, but they're honestly way too long together, so that's why they're separate.